press record now so that everybody is. So for those people online, welcome. This is week 2, the 1st of March 2018. We are starting today's class with a DIY prac. So I'm going to turn you around and hopefully you can see okay, can't you? Right. Excellent. Say hi to everybody. Everybody say hi. So just to remind everybody, we are online now from now on, so that the people that come in remotely can access these classes. It is better if you can turn up because it's far more interactive because you can get to stop me and ask questions. But that said, it's better than missing the material altogether. So I'm just going to close up the screen so that we don't hear everyone in the corridor, and then I can make a start. Okay, so welcome to week 2 of Agronomy 1, the subject that teaches you all about how to grow plants commercially. I want to start this by overviewing the DIY practical. So I emailed on Moodle last night a copy of the DIY practical. You can hear me okay, can't you, Manishka? Yeah, cool. And I also put it up on the assessment section, which I don't think is quite open for you yet, because I wanted you to be in at least week 2 before I scared you before I get assessed. But you do have the student subject outline, so you do know the overviews. So in the DIY practical, it's split up into 2 practicals. There's a seed germination, which accompanies the plant growth and development topics. So in other words, what I hope that you get out of that particular practical is to see plants, 1, germinate, 2, to begin to understand that in a commercial context, which is the part of the assessment. It's not that you're counting and looking at seeds germinating, it's that you're actually taking what you're seeing and imagining it in a field setting, because that's really important, because that's the first step of your understanding. How do plants get established in commercial settings, in paddocks? So hopefully these treatments will give you some idea of how that works. So what you'll do today is you'll collect 2 different seed varieties, which most people have, and we'll make sure that everybody's made a note. So I have a note. So in theory, the whole class should have compared a different set of species and treatments, because that will, if we then share our results, that will give us a lot as a class to discuss and review when we come to that part. So in your DIY kit, you will get this stuff, which is called parafilm. Have you all used this before? It's like sellotape. It's a very stretchy material. It's very expensive, by the way, and it's made out of silica. And it comes with a paper side and also a very waxy side. And you can just make a slight nick in it at the top, and you pull the paper off, and then you get a see-through sort of stretchy plastic. And this is what we use to seal the Petri dishes. So this is your first component, is your parafilm. Your second component is a Petri dish, of which you will hopefully have a number of seeds to place in. The next component of the experiment is filter paper, and that is used because 1, it's clean and sterile, and you'll all get a piece of filter paper. I recommend putting 2 pieces of filter paper onto your Petri dish. Now, some of the filter papers work well for size, and others you'd have to cut down. And the cutting doesn't have to be perfect. It just has to fit in squarely at the bottom of your container. So what I tend to do is I just use the base of the Petri dish as a guide to cutting the approximate size down like something like Blue Peter on us on kiddies program now. Okay, so you should have your Petri dish is nice and flat so that you can lay your seeds on it. So that's step one, put your filter paper into your Petri dish. Step two is add about a teaspoon, maybe 2 teaspoons of water. And it really is a judgment call for how much water you need to add. So I add a couple of drops at a time. And what you will see is you'll see that there should be enough water just for it to be moving around, but not too much so that the seeds are floating. So I'm just going to add a fraction more. I'm just going to show everybody what that kind of looks like. So you've just got a tiny bit of water moving around. And what you're trying to do is you're trying to make sure that your seeds come into enough contact with the water. If your seeds are very big, guess what? Well, it's for your particular scenario, because you've got big beans, I would definitely get bigger filter paper. And for the beans, these are the broad beans, I would probably have at least 3 pieces of filter paper for that particular species, because they require a lot more water. Well, it's to do with the surface volume ratio of water seeds take up. If you're a tiny seed, you don't need much. If you're a massive seed, you are going to need more. All right. So the next step is to place your seeds into your Petri dish. Now, what you're looking for is a nice coverage over your Petri dish, because if they're too close together, you can get a phenomenon of inhibition of germination for some seeds that promote that. But also, if they're nicely spread out, it's much easier to count. And what you're going to do is you're going to monitor your seeds once they're set up every day or every other day, and you're going to count firstly how they germinate. So when they, does anybody here know what germination actually is? Come and join us. Germination is the breaking of the seed coat, and where the radical, radical first appears. All right. So if everyone can see online, I have got this set up here. Sorry, I'm not doing a very good job of the camera, but there it is. Can you see that online? How it's nicely spread? There you go. Hopefully you can see. It's not brilliant, but I'll take a picture. And for you guys in the room, hopefully you can see, it's reasonably spread. So then what you do is you get your lid, and at this point I would take a permanent marker if you have one to write on it, so the variety. So I would, in this case, we've got, this is fescue, isn't it, Grace? You chose fescue, it's your second one, yeah. So we've got fescue, and what you do then, and this bit is a little bit tricky because you want to keep the Petri dish flat, you don't want to tip it because otherwise the seeds will tip. And then what I do is I have a thumb on the top of the dish and a finger underneath with my index finger free, because this is the technique I've established. And then I put the parafilm on, and then on one corner like that, and I just hold it with my index finger. You'll see that? So like this. And then very slowly I pull the parafilm over. Now, if you go too quickly, it will break. If it's too cold, it will break. Usually, it doesn't break if it's too hot. But then, for those online, you can see, hopefully you can see that. You can see now that that makes a beautiful seal. So what you want to do today is you want to take 4 pieces of paraffin and then I guarantee the first time you do it, you'll break. Just because, and be slow with pulling it. Don't be quick. That's the trick here. See that? So your fill. You need to choose. I would recommend or you can do them, but we've got the best for you. You don't have to do it. Also find folks.
पर AGR से चोर लो.
So, I mean, you know, you're more than welcome to do this.
वो AGR से मुंह फुलटा में.
That's very relevant.
AGR से चोर.
I'm sure
Fortnite.
all right. So, are we all clear? What we do? So, does everybody want to go and get the rest of with your filter paper? And I'm just going to pause the recording of this because we don't need to. Yeah, cool. So another thing to understand, guys online, is that these seeds are not for human consumption. So they're not designed to be eaten. So any small inquisitive children in your life, keep them away from the seeds. Some seeds have inoculations on them as well. The seeds germinate and they can help also getting the seed established. We'll learn a lot more about that when we visit the Wilson Seeds Research and Seeds, because they do a lot of inoculations on their seeds. So I'm going to touch on that today. I'm not going to touch in any great detail. HUNDAI. Okay. I'm going to pause. Okay, I'm going to talk about treatments now. And hopefully you can all see this. In your DIY practical on page 7, there is a list of treatments, and the first one I've called the control, and that will be at room temperature and it will be in the dark. So you can just put it on you know, just somewhere there it's not going to get disturbed. So it can be covered if you want, or it can be on the, you know, on a desk or on top of the fridge, or not top of the fridge. But one thing that I will give you all is a piece of foil to wrap your dish in. So why? Can anybody tell me why the control in the dark? Why is in the dark the control?
Actually, okay, it's the other way because it will look at temperature main plot.
So the objective of this practical is for you to begin to think about commercialization of fruit farm, commercially that is. So what I would suggest to you is that most commercial growing situations are that the seed is put in the seeder, and that seeder doesn't just spread the seed on top of the soil. Most seeders will actually turn the soil over and put the seed under the ground. Or there are some seeders out there that you lay the seed on top of the ground and then you sprinkle soil on top. But that's the more specialised industry. So in most situations, in a commercial situation, your seed will be at room temperature or whatever the temperature is in the dark. Agree? So what you then have to think is what treatment you would like to expose your seeds to. I think I'm going to give you 2 pieces of paper, but enough for both. Is it enough foil to cover both? You think? Yeah, more? Yeah, no, nothing special about it, yeah. Yeah, heaps at home. There was a hole in one piece of yours there. So hopefully that covers that. Oh yeah, that definitely will be yours. So what you'll do is your first species. So let's say your first species is fescue. You'll put that, you'll wrap that in foil, and the same as your second species. So you'll have 2 Petri dishes wrapped in foil, 1 from each species. Okay, so the treatment that you choose, you need to put both of your other Petri dishes on that. So if you've got fescues and oats, you'll have fescues and oats in the dark, 1 Petri dish, and then you'll have fescues and oats in your treatment condition. So your treatment can be in the light, at the same temperature ambient as your control, or your treatment can be in the dark, but in the fridge. So that would be about 4 degrees. And I've asked you to make a note of the approximate temperature. I don't expect you to go out and buy a data logger temperature every minute. Although if anybody wants to do that, that would be quite interesting. I just want to know, most homes are usually about 10 degrees if they're not air conditioned or heated. I'm assuming some of you all live in typical student type accommodation, as I did. So probably 10 degrees below ambient, whatever the weather says it will be, is usually what is in a house or a home. If your home is auto-controlled, most people set that around 21, 22, 25 degrees C. So if you can make a rough note when you're writing this up of your temperature. The other place that you could do it is you could put it in the dark. So you could grab another piece of foil and then put that on in a warmer environment. So you don't want to cook it, obviously. Don't put it in the oven because that won't go well for your Petri dishes or your seeds. But a place that is very typically warm is on top of your fridge. Just make sure it doesn't cover an air vent, for example. My fridge is always warmer than the rest of my house if I put my hands on it. Or you can put it on a windowsill, wrapped in foil that gets a lot of sun. So if it's on, the west side of a house, say, here in Australia, that will be quite warm. So you're quite clear? Does that make sense? And what you're going to do is every day you're going to monitor it. You're going to watch. The amount of water. If the water starts to evaporate, then what you need to do is replenish it and then refill it. There will come a point when your seeds will germinate, and they won't all germinate at once, so you'll have to go back every day once they start germinating. And you might see in the first day 2 or 3 seeds germinate. Then the next day you might see 10 seeds germinated, and then the following day you might be up to 18. You might not get 100% germination, which means that not all seeds may germinate, and that's just part of the experiment because that's what happens. Once you've collected that data, you'll be able to calculate your germination rates. So you'll see how well and your germination and your viabilities of your seeds. So if your seed is a really good seed, you'll have every single seed in your batch germinate, and they'll germinate quickly. Why do you want, from a commercial perspective, seeds to germinate quickly?
Get quicker crop, maybe?
Say that again, Mustafa.
Get quicker crop.
So why do you want a quicker crop? Why do you want the crop there quicker?
To sell in the market for.
So if you invest money in seed, every single seed that doesn't germinate is costing you in theory, right? So that's one reason. The second reason is that when they're in the ground as a seed, there are predators like birds and insects in the soil that will eat them, given half a chance. So the longer they take to germinate, the higher the risk, as a rule, of them being non-viable, shall we say, either attacked or not being used. Okay, once they've germinated, once most of them or all of them have germinated, you'll start to get the first bit of growth. And usually the coleoptiles are the first structures in the plants that you will see. They're not leaves, they're coleoptiles. And I'd like you to allow one of your treatments from both species to grow a little bit so that you can see it develop slightly. So I would hope that you would, once they've all germinated, you remove the lid of the Petri dish and add a bit more water. You'll need a fair bit more water now that they've germinated, but don't soak them in it, because if they float, what will happen? Does anybody know? Why is that not a good idea if you have too much water? They actually need air, and so the water stops the air. It then turns to what we call anaerobic conditions, which can then cause all sorts of problems. It can cause diseases to cause, and you may find this. You may find that your seeds get diseased, and that does sometimes happen in the soil environment as well, and you can lose seeds to soil, to disease, which is why sometimes inoculation can help prevent that as well. All right. And photographs. If you've got a smartphone or something like this, you can take some photos with. That would be great. Okay. So, and I suggest that you also, at the start of the experiment, take note of the seed size. So have you got a very small seed, or is it quite a large seed? Because we have a spectrum of seeds there. What is the colour of your seeds, and how did they feel? Did the seed coat feel very tough, or did it feel weak? Okay. All right. So are we all clear on the DIY experiment? Because I'm going to move on now. Excellent. So now we're going to look at the lecture 2, which is finishing off the crop physiology. I'm just setting this up so it comes on both screens. This takes me a few minutes. Guys online, can you see the handouts? Okay, just nod. Yep. Excellent, excellent. All right. Oh, I've got the wrong lecture. Sorry, that's the second lecture. Lecture 2 is what I need. Crop physiology. Yeah. Great. So we're going to continue the crop physiology lecture, topic, sorry, and this is lecture 2. Now, in the first lecture, we looked at photosynthesis, and we also looked at some of the carbon metabolism classifications. Today, we're going to concentrate on plant growth and development and something called phenology, which I'll explain as we go on. This is a very basic lecture. It will bring everybody up to the same understanding and level as you all have slightly different things. So I guess the first thing is to contextualize, why are we studying plant growth? You know, and it's a very simple reason, because it's really, really important from a commercial perspective. You need to be able to understand how a plant grows and develops in order to manage it, because to manage it optimally means that you do certain things at certain times, basically. And if you don't understand what that time is from a plant's perspective, then you'll get your management wrong. So, for example, if you're told to add nitrogen at EL12 stage, and that's what your agronomist does, and he's consulting with you, and then you go away and you go, then you're going to miss the boat and you're not going to optimize your crop return, for example. So this lecture and some of the exercises that we will do both now and later on in the subject are all about you becoming very familiar with the basics. And I cannot emphasize how important it is for you to know what these plants look like even. You know, it's really, you know, you can't go and work as an agronomist or work in ag if you don't know the difference between a plantain and a fescue and a broad bean. I mean, most of you will have a basic understanding of them because you eat these foods and you come across these foods. But what I hope by the end of this subject is that you'll be able to walk into a paddock and you'll know what canola looks like. You'll know what lucerne looks like, for example, okay? So if we understand plant growth, we then have the potential to optimize our commercial inputs, and thus maximum productivity will hopefully be gained. And if we have maximum productivity and we've optimized our inputs, then we get the best economic return or profits that we can. So that's the aim of this, really. So plant growth measurement plays a key role in optimizing economic return. Now, I don't mean currently that farmers go out and they spend hours and hours with a ruler measuring their plant height. They don't. But we are very close to getting technology that can do that. So perhaps in the future, that will start to happen. Mostly with plant growth measurements, we use, it's utilized in science. And so we measure different components about the plant, and we will compare different commercial ways of growing that plant. And then if we understand the development and the growth, we know exactly what to measure and how to measure that. So some of this subject, you will be learning some of the very basics about plant growth measurement so that you yourselves can conduct on-farm experimentation. But most importantly, you can understand what the literature is meaning. When it says leaf area index, what does that mean? Does that mean anything to you now? No? No? Plant height does, yeah, that's quite obvious. But hopefully by the end of this series of this subject, and certainly by the end of plant physiology subject in year 2, you'll have a very solid understanding of that. So I want to start looking at the basics of growth. Now, you'll cover this in living systems in far more detail, so I'm just going to touch on it now. But what makes something grow? So we have cell division. That's one form of growth, isn't it? So you get one cell and then it divides into 2 cells. Yeah, very simple. That is growth. Once the cell has divided, the next stage is it expands, right? It becomes bigger. That is also growth. The third stage is that we call differentiation, which means that it becomes functional, that it signals a function. So what is that plant cell going to be? Is it going to be a part of the photosynthetic mechanism, or is it going to be part of the cuticle, or is it going to be a stomata? We require protein synthesis for growth. That's a very important component of growth. And also growth is irreversible. So you don't go backwards with growth, or at least from the definition of a plant perspective, you don't. And largely, another component to understand about growth is you need energy to do that. And as we learned last week, photosynthesis is the plant's way of obtaining energy from the sun. So we're going to get into the nitty-gritty of cell division and then expansion a little bit here. Again, you'll do this in far more detail in living systems, but I'm just going to touch on this. So cell division is the process by which a parent cell divides. So it's very simple. You have a parent cell here, divides into two. It can divide into more than two, and these, the cells that come from these dividing cells are called daughter cells. Plants, unlike humans, have the capacity for unlimited growth throughout their lifespan. So that is a very important difference. Growth can be divided into a number of phases. So the first phase that we have is called meristemic. So that is usually occurs in the meristem as well. That's not a coincidence. The meristem is where meristemic growth occurs. You then have cell elongation, and you then have a maturity or maturation. Meristems are physical regions where the plant grows. And this is something that is very important from a commercial perspective, because if you cut the meristem on a plant, it's game over. So this is why things like grazing, we'll spend a lot of time when we do pastures and understanding how long an animal should be in a paddock, because if the animal eats too much of the grass, it can get down to the meristemic region. And the minute it eats the meristemic region, then there's no way of the plant dividing. That can also be very useful knowledge for weed control. Sometimes if you've got a paddock that you want cleared, you'll go in and you'll put cattle in there and or something that really eats really close, and if you can get as much of the meristemic region of the plant eaten, then your weed's gone. it'll shrivel up and die, and that'll be the end of it. Okay, growth at the cellular level is principally an increase in the amount of protoplasm. So that's actually what's going on. What I want you to understand is that growth can happen in a number of ways. There's what we call geometric growth, and there's arithmetic growth. And they will depend on how the parent cell divides. And so you get different types of growth depending on whether your cell undergoes arithmetic growth or geometric growth. And this slide here, if I can take the pointer, sums it up quite well. And you can see the difference in cell number. So from a arithmetic growth, in the first division, you'll have 2, which is the same as the geometric. But in the second division, you'll have 3. Then 4, then 5, then 6. So if you've gone through 1, 2, 3, 4, 5 cycles of division, you get 6 cells. Geometric is where you divide in 2 through each cycle. So you go 2, 4, 8, 16, 32. Which one of them has a better growth rate? Geometric, yeah. It's a simple and obvious question perhaps, but that's what it does. Are you guys online quite clear about that?
Yes, Nikki.
Yep, excellent, okay. All right, maths, who likes maths? Yay, excellent, we got a little tiny bit of maths. These are the formulas that represent arithmetic growth and geometric growth. Now, when you're a plant physiologist or plant scientist, you'll use a lot of formulas to calculate different perspectives of growth of a plant. And this is just introducing you to the first component. I don't expect you to learn these formulas, but I do expect you to understand approximately what they're doing. So we have L, in this case, LT equals the length at time t. So we're looking at the length of the growth. The length of time at t0 is indicated by L and then a 0. And R is the growth rate, where lowercase t represents the time. So if you have the length at the beginning of your measurement plus the growth rate multiplied by the time, you then get the total growth or length. Geometric growth, as you can see, is a bit more complicated from a formula perspective. This just involves adding up a multiplication, whereas the geometric growth also has adding up, but there's an exponential component, and that's because you go 2, 4, 8, etc. So that's what that's indicating. Does that give a clear component or a clear understanding of the differences of these two growths? Yeah. So what should we measure when we're measuring plant growth? What's important to us? Well, the fresh weight of a plant. So how much bulk does that plant weigh? So if I was to grow, let's say theoretically, I keep meaning to bring a plant to class, but I don't have any on me at the moment. Normally I have a glass full of them, but I like to use a plant. Imagine this is a plant, and these are the roots, this is the stem, and these are my leaves. The fresh weight is taking that out of the ground, washing all the soil and debris from it, and then placing it on a balance. And that gives you what we call the fresh weight. That weight will include all of the water that is in that plant at that time, and ideally no debris. Now, in the early 30s, the 1930s, the 1940s, there was an awful lot of science around how you should measure plants. And what they found out is that within 1 to 2 seconds of removing a plant from its environment, so if it's in the soil, 1 to 2 seconds, that plant starts to lose water. At the same time it loses water, there's a whole suite of cell signaling that goes on where the plant goes, uh-oh, this is not good. It recognizes that it has been moved, or well, what it recognizes is physical damage from being removed. Plants are very good at recognizing that, and then they upregulate, and that's the fancy, they turn on a switch of genes, which then means that they can elicit some defense mechanisms. So when you do fresh weights, if you're being highly scientific about it and you're in the field, you do not have very long to take your plant, clean it, and weigh it. And what we tend to do is acknowledge that as an error, but we do it systematically. So we take the same amount of time to measure the plants across all of our treatments. Okay, so that's how we do fresh weight. Dry weight, can anybody guess what that is? Yeah, you do. And there are very standardized methods for doing that. So we have commercial ovens here at Epping for just doing that. You can also use microwaves. And it's a bit like cooking. You have a set amount of time that they need to be in the oven at a set temperature. Now the temperature has to be warm enough to remove all the water, but not too hot, so that it degrades everything else. And the plant, everything else is made up of proteins, it's made up of enzymes, it's made up of carbon molecules and everything else that constitutes that plant. Some components of the plant are gaseous. So, there are the parts of the plant that are gas that you will lose. So there's a hormone that's a gas, for example, that you will lose in drying. And if you were interested in that particular hormone, you obviously wouldn't do a dry weight because you would lose it. So you'd have to find another way of measuring that. Usually we do labeling experiments, and then we measure either the radioactive presence of which has a relationship, or we have some kind of dye process. The stronger the dye or the color, the more of that molecule is in the plant. So dry weight, very simple to do. One of the things about dry weight is that the material is what we call, plant material is what we call deliquescent. Do you know what that means? It means it will reabsorb water. So if you're doing this for a scientific perspective and you want to know what all your treatments are, and you put them in the oven, let's say you do it for 24 hours at, let's say you do 68 degrees C or 72, that's quite a common temperature depending on what plant you're drying. You then lay it out on the bench, and it's a very hot and humid way, and you think, oh, I know what, I'm going to go and grab a coffee first. and then I'm going to have lunch, and then I'm going to have a chat on my mobile, and then I'm going to come back into the lab an hour and a half later. Chances are that all the moisture in the atmosphere, that plant has reabsorbed, because plants are really, really good at absorbing water, so they're delicate. Okay, so that's just something you have to be mindful of. If you're measuring plants, you've taken the oven, you need to sort of process them quite quickly. Other components that we can measure are the length or the area, the volume, and cell number. Cell number is ideal, but extremely difficult to do. And basically, you have to take sections, you have to stain them, fix them, and put them on a microwave, and then count them. And then you apply a formula. So if you've counted a number of sections, that represents the percentage area of that particular organ or component of the plant, and then you multiply that up to give you the final cell number. It's quite difficult to do that. I want to talk you through very quickly a sigmoid growth curve, because this is what is most typically found with plants, and this is how they tend to grow. So what we have here, this DM axis on the left-hand side of this graph. This is so exciting, having a graph in your second lecture, eh? Or third lecture. So we have DM, which is dry matter. So in other words, our plant material has been taken out of the ground, it's been put in an oven, and then we've weighed it, ideally to at least 2 decimal places. And this is time. So what happens initially? Nothing, if the plant is growing. Then over time, it will slowly begin to increase its growth rate. And this represents the first processes of cell division, right? So you've got the parent cell, then you've got the first lot of daughter cells, and then they have daughter cells. And so it's quite slow to begin with. But because we're multiplying and increasing cell number, it becomes exponential or rapidly increases in the section B. And that's called the log phase. So you can see that, can't you, going up very, very quickly. Then it begins to slow down here. And then at D, it just stops. And E isn't, in fact, growth. What is E? Not a happy day for the plant, yeah. E, the technical term is senescence, when a plant naturally dies. So it senesces, then it dies. Senescence is an extremely interesting section of plant physiology, which you'll look at in plant phys lectures. But it senesces or begins to die, and eventually it will die. Now, this is a typical shape, but you can change the shape of this curve by the inputs that you add. And so this is something I really want you to take on board. Climate can change this, light can change it, temperature can, water can, day length, and wind. And all of these factors I've just listed could change it positively, but also negatively. So let's talk about a couple. Light. Does a plant need light to grow? Why? Good, photosynthesis, yep, excellent. So can you give a plant too much light then? Yes, well, that's an interesting one. They have done a lot of 24-hour experiments, and it seems from a photosynthetic perspective, as a rule, no, but there's a whole load of timing mechanisms or circadian rhythms in plants that get thrown out if you give it too much light. Of course, in natural environments, unless something really spooky happens to the world and the planet and the earth, we have natural cycles of day length. From an experimental perspective, we use either one of two. We either use 8 hours or we use 16 hours of light, because plants tend to fall in 2 categories. There's the short day plants and the long day plants. I'm not going to discuss them here other than to mention that they exist. So light can impact it. Temperature. Is temperature a good thing or a bad thing if you're trying to optimize productivity? Excellent. And the species. So some of the plant varieties we've looked at today prefer temperate climates. There are, I believe there's at least one in there that prefers more tropical climates for growth. So that may impact on your experiment that you're doing at home. So yes, temperature can be positive or it can be negative with respect to dry matter and carbon acclimation. Wind, wind, not enough wind can be a problem, and too much wind can definitely be a problem. Too much wind has a physical issue. Too much wind can cause too much of an evaporative demand. Evaporative demand is the process where the plant takes water through itself. So the plant doesn't actively, well, it can actively, but mostly the plant will, it'll use the physics of the evaporative demand. So how much negative pressure is in the roots versus the negative pressure in the atmosphere. And the bigger the difference, the quicker the water moves through the plants. Again, we're not going to learn about that in this subject. We'll learn about it in plant physiology. So other factors that can influence our growth in plants are the soil type. There are types of soil which don't grow very well for some plants. There are other types of soil which are excellent for plant growth. So let's take Gippsland. Is that a good area for plant growth from a soil perspective? Gippsland. Do you know Gippsland? Dairy country? Yeah, Gippsland, I mentioned it's because one of the best soil types we have in and around mainland Australia. So it's really, really good, productive for most, not all, for most species. How about if we go further north to somewhere like Mildura, north and central? Yeah, no, well, it depends. It's good for carrots, sandy loamy, good for some grapevines, for some rootstock selections, good for asparagus, a lot of asparagus growing up there. Not so good for some other plants. All right, not so good for wheat, for example. pH of the soil, which you'll learn about in ecological systems, and you'll learn a little bit more in the soil lectures a bit later on, so I'm not going to touch on that, but that can definitely influence how a plant grows. It influences the plant because if it's too acidic or too alkaline, it can damage the plant roots. If you damage the plant root system, you're reducing the amount of water and nutrients that can be taken up at any one time. It can also interfere with the chemistry about taking up nutrients, and that plays a very important role. So if you haven't got your nutrients in your plant, then you can't grow and develop, obviously. Are you guys online or good? You're all getting this? Yeah.
What about the day length, Nikki? How the day length is?
The day length? Yep, the day length can impact. So there are internal responses for plants. So you can have short days and long days. If you put I have to remember which way round it is, but I think you can put short, I'm going to have to check, but one of them you can't put it the other way round. So it's like you can't put a short day plant in a long day or vice versa. I'll have to check on that, I can't remember, but because it disrupts the chemistry for the hormone production. Yep. And that signals a whole load of other processes. So it's the disruption of the circadian rhythm that tends to be a big driver for day length. But in a simple sense, if you have 12 hours of daylight, in theory, you get more productivity than you do if you have 8 hours of daylight. Okay? And if you are setting up chamber experiments, that's also important as well to do that, understand that day length.
Daylight saving will affect plants?
No, it only affects humans.
Really?
Mm.
What's the daylight that because obviously during daylight saving you get longer sunlight.
Okay, so the length of daylight you get is set by the way the Earth rotates around the sun, very basically. And we as humans put an interpretation on that called time, or the monitoring of time. So the time of year that you're talking about from a daylight savings perspective is, yes, you will get more sunlight at that time of year, but the changing of the clocks, or what we call daylight savings, is actually an interpretation of that time. So you get more light during the summer months as a rule in most countries. There are some exceptions, of course. Does that make sense? But the change when the clocks go from in daylight savings, you know, from 1 hour forward or 1 hour back, that's a human thing. But a good question, good question. Yes, plants follow The circadian rhythms, there's the science behind circadian rhythms is absolutely fascinating, and there are a suite of genes that are turned on and off regularly throughout the growing cycle of the plant. So the plant knows when it's in the day and when it's in the dark because of its circadian rhythms, and there's been experiments to disrupt that and bring it back. From a commercial perspective, you need to understand that light has an influence when it's cloudy, mostly from Australia's perspective. So we normally get excellent light here in Australia. We normally get really good spectrum. So we don't get much light pollution, unlike the places in Europe that can suffer from light pollution. So the plants grown here get a good blue, they get good red and good far red, and that tends to be mostly where they capture the energy from. On cloudy days, you'll get less light. If you get less light on those cloudy days, you'll get less photosynthesis. Less photosynthesis means, as a rule, less productivity. So yeah, and there have been seasons where we've been very cloudy over the summer here in Australia, and you definitely see the difference. Can you do anything about it?
Visualize?
In most situations, no. Yeah, in protected cropping, you can possibly, and sometimes they do, but as a rule, no, we don't. Okay. Other things that impact this growth, obviously, are nutrients. If you have all the nutrients you need, your growth will be optimal and look something like this. If you're limiting on one nutrient or more, then your growth will not reach optimal. So there is a concentration and quality component to your concentration for your nutrients and quality for your growth relationship that occurs. Weeds. Weeds impact because why? What do weeds do? That's right, especially if they're trying to get established. They do cover up the plant. That's a good point. What else do they do? Excellent. Yes, they do.
Is it sharing your food?
Yeah, really good. So they do compete for nutrients or sharing of the food, and yes, they do compete for all of the components. So nutrients they compete for, but they also compete for water and, as you rightfully said, light as well. Okay, insects. They can be harmful because they physically eat the plant. Diseases, we have many in Australia. You will learn lots about them in the course of your time in agronomy. Nematodes. They can be physically damaging. They can restrict root growth, and soil microbes. Soil microbes can be positive and they can be negative. The wrong microbe community that's allowed to get hold can be very negative. A well-balanced ecosystem containing a mix of microbes and fungi are absolutely excellent because they enable nutrient cycling in the soil, of which you will learn more about that soon. There are other internal factors that can, or what we call genetic factors, that can impact on plant growth. We have resistance to stresses. We have photosynthetic rate changes, changes to respiration. So some stress, some climates cause plants to respire at different rates, which then can cause issues. And another component that can change your signal growth curve is how what we say, the technical thing we say is how your carbon or your assimilates are partitioned. So that's the fancy word for saying where is the carbon going in your plant. We use the word carbon because carbon makes the backbone of all of the compounds in the plant pretty much. So if you're a cell wall and your cellulose, you've got carbon in them, you've got carbon everywhere. So do you know what's the most common element in a plant? What?
Carbon.
No, carbon's very close, but it's not the most common. Nitrate? No. Ooh, very close.
Water.
Water, yes. So usually water is about 70 to 80% of the plant weight on a weight basis. So when you start to, when you have a fresh weight measurement of a plant, you're including how much water is in it. And that is very influential as to the time of day that you've collected that plant. So, you know, how much, if you've collected it during the day and it's a hot sunny day, then it will be on the lowish side of water. If you've collected it at 5 a.m. in the morning and it's had a really good watering overnight and it's not under stress conditions from a water perspective, then it should have a lot of water in it, relatively. All right. So we've looked at plant growth. We've now understood that it constitutes cell division, elongation, and differentiation. We've looked at specific stages of plant development within the plant's life. We are now beginning to understand that the rate of growth is important. It's important from a commercial perspective, and it's very much influenced. It's influenced by some things we can control and some things like the weather and climate that we can't control. So do growth and development proceed at fixed rates? That's a question for you.
No.
What do you guys online think?
No, because there are a number of factors like depending on like wind, temperature.
Yep, that's right. So growth and development doesn't proceed at fixed rates all the time. It changes, and we have optimal rates. So we do understand if everything is in the right way. In other words, if the nutrients are optimal, if the light is optimal, if the wind and climate and temperature and humidity are all optimal, then there is a maximum growth rate. But anything can influence that. It can make it positive or it can make it negative. And just touching finally that on this topic about development, it's modified by environmental factors, which we're beginning to learn about, and we will learn about in a bit more detail. But there's also management factors that can influence how your plant can grow. And your management factors can promote plant growth, but it can also, if you get it wrong, inhibit plant growth. So an example of that, let's say you're growing pastures, and let's say you put your horses, your sheep, or your cows too long on the pasture. Then that would be a negative consequence on growth. I mean, it's negative anyway for a short time because the cattle and the horses are eating the grass. But if you allow your cattle and your horses and you have got good rotations, then that eating will promote new growth, and then it will be ready for the next, and it will be growing, and all of the resources will be optimized through that growth. So now we've understood the basics of growth, I want to touch on development. So do you know how growth is different from development? I mean, it's on the slide. Yeah, it differentiates between the various stages of development of the plant. So what you're going to see in your DIY experiment is seed germination and early development. What you're hopefully going to see out in the farm later on in the subject is that you'll see different stages of the plant growth, and we call those different stages the phenological stage. So is that a term any of you heard before? Any of you guys heard that before?
No.
No. And I'm going to start with wheat, because we always start with wheat because there's the most written around wheat, and once you understand wheat, you will understand the rest. So each of the different stages has a coding. And the reason that we code it is because we can then align our commercial practice next to those codes. So in other words, the first stage is pretty simple in what you'll be learning about over the next few weeks, and that's germination. Germination is the splitting of the seed coat and the appearance of the first structures from that seed. This is followed by emergence. Then you have tillering, which is the next stage, and that's where a lot of the sort of green type structured growth grows, where the stem is actively growing and you're getting more and more stems and more leaves. Once that wheat plant has hit a certain maturity or it gets too much stress, it then goes into a floral initiation. So that's the start of the flowering process. You then have a terminal spikelet, and that describes how the flowering process actually looks, and it's defining the stages on the looks. Then you have the first node or beginning of stem, then you have boot, spike emergence, anathesis, and maturity. And the four stages listed are E, GS1, GS2, and GS3. These stages are very broad stages, and within them there is set stages. So let's have a look at the stages within them. So what you can see here is we have sowing, so that's what we're doing in our Petri dishes. Then you have germination, and that occurs at this point. Then what we have is emergence, so that is the plant. You start off with the coleoptile, then that will develop into, then the leaves will start to emerge and stem structures will form. And this is where we're beginning to you'll get your first couple of leaves at this stage. Then you get the plant growing more and more. And this is this stage here is what we call tillering. So you're getting tillering means that these stem structures are growing and the plant is splitting up in tillers. So when you first get your wheat seed germinate, you'll have one stem shoot up, and then at the basal meristemic area, you'll get more cell division, which will cause another stem to come up. Each stems have a number of leaves that come up from the base. This is a very defined and very specific way of growing. It typifies the type of plants called monocots. Dicots grow in slightly different ways. Wheat plant is a monocot. Fescue, grass, is a monocot. Okay? Beans, are they monocots or dicots, do you think? They're dicots, yeah. And again, you'll cover this in Living Systems 1. They grow quite differently to a monocot. But this, all we're describing here is the monocot. So as we get tilling, and then you get floral induction here. Here we have flowering, and here the crop and the wheat seeds are ready to harvest. All right. Now, there are recommendations for when you add nutrients and across all of these different stages, and this is why it's really good to understand this. So we would put some nutrients in at sowing, but we would keep the concentration relatively low. Do you know why? Because one, if the concentration is too low, you can burn. Have you heard of nutrient burn? You can actually burn the seeds. Two, rain will fall and a bit of nutrient will get washed out. The plant will use a bit of nutrient. A bit more time goes. More rain will fall. The plant will use a bit more nutrient. Rain will wash some of it out. Some of it might do what we call volatomize, which means go up into the atmosphere. So in other words, even if you do put all your nutrients on at the start of your growing season, and even if your seeds aren't burnt by having too high a concentration, as time goes through, as goes on, some of that nutrient you'll be losing to the atmosphere, or it will be flushed through the soil and your plants won't be able to get it. It's not what you add that's the most important component, it's what the plant is able to access that's the really most important. So this is why if you then put certain nutrients at this stage and then more nutrients at this stage, you can really optimize your growth. Now when you're growing wheat for things like noodle production, you will feed or put nutrients on a very different way than if you're just growing wheat for fodder. Why is that? Do you know? Because you get a lot more money for top grade wheat for noodles than you do for wheat for fodder. So in other words, you don't want to keep putting the tractor over it. You don't want to keep putting nutrients on it. If you can get away with a very low input, if you've already got X amount in your soil and you know that, that's your best practice, then that's how you might manage the same crop but differently depending on the commercial output. So you know how I was knocking on about the word optimization last week. And I want you to move away from things like best, because it's not a very good descriptor for this type of thing. So we optimize our inputs, in this case nutrients, for our commercial outputs. And they'll be quite different from a noodle production than they would be for wheat for fodder. And that's a very extreme case, but that's what we tend to do. And if you understand the phenological phases, you can optimize that because you're putting your nutrients on at the right stage. Here are the 3 stages. This represents the approximate lifespan. So E is quite short, GS1 is longer than E, and GS2 is probably the longest, and that's where most of the vegetative component, and then this gives the time for GS3 relatively. What we have here is a table that summarizes for wheat the physiological stages. So we've got days from emergence to physiological maturity in spring and winter. So this is the spring data and this is the winter data. Can anybody tell me what the difference is? What do you note from that data? Anybody online? Takes longer, did you say? Takes longer in winter. Yes, it does. Yep. And why do you think that might be? Yep. That's right. And temperature? Yep. Temperature, light, really good factors. So when you're managing your commercial entity, you have to understand that when you sow or when you put plants in the ground, that will alter their productivity and their processes. And so that change will speed up, will shorten or lengthen these stages depending on what the factors are. All right, so is that clear? Yep, okay, cool. So we've got from seed to sowing time is the duration of your crop. In a pasture, that's not quite the same. It's from seed until you remove your pasture. That's quite a lot longer. And the phenological stages in pastures, we tend to manage so that we stay at a vegetative stage. We don't tend to allow the plant to go into flowering. How do we do that? How do we keep the plant in a vegetative state? We put the animals in it, right? We have a feeding rotation. Okay. If a crop is usually grown in winter, and if you sow too early, you'll miss out on the rain in Australia. And if you don't have rain, then it won't germinate, or the germination rate will be low. Even if it does germinate and it doesn't rain, the amount of water in your soil might be quite limited because you've just come out of summer, right? And if it hasn't rained a lot, then that can be a problem. Although summer rainfall in Australia is changing, it's increasing. And smart farmers have realised this and they're starting to grow summer crops to utilise that rain. However, if you do grow a summer crop, you have to remember that that's going to change what you might do in the winter as well, because you won't have any of the holding capacity because the summer crops move the water out. So it's always a bit of a balance, always a little bit of a give and take in your farming. If you plant your winter crop too late, you may get enough rain, but then the temperature gets too cold and it takes a lot longer. It won't stop the plant germinating and it won't stop the plant getting to maturity usually, but it will slow it down quite considerably. And of course that has impacts at the other end. So if you're not taking your wheat off till late November, and suddenly it's really, really hot, you could lose productivity because your wheat is at the wrong end of when you'd ideally like to harvest. which is before then. Okay, so is that clear how phenology and timing can impact your crop productivity? And you know, as farmers, you spend a long time thinking, when should I put this crop in? When should I establish this pasture to get the optimum output? If you're going to intentionally put in crops early, then you need to put varieties in which are adapted to early sowing, and these are called early varieties. If for some other reason, there's other reasons that you intend to manage your crops so they go in late, you want varieties that are okay being late sown. So you can get varietal differences that help buffer some of these differences. And so varietal selection is really, really important in commercial crop production. Okay, so that leads me on very nicely, because I set that up to do so, is to have a quick talk about the assessment, which you all got a copy of last week. I did. Did you? Oh, did you get a copy of it last week? Oh no, maybe you'll get a copy of it now. I will email it. Sorry, I will put it on Moodle and open up the assessments today for those that don't have it. But this is for agronomy students only, so you can close your eyes for a moment and you read. I can copy out more. I don't know how many times you've got enough. But what you need to do, Staff has already done this, is that you need to select a plant, but you need authorization. You don't select. I'm going to have to print out something more in breakfast. And each person, you've got one? Each person has a theoretical crop, and what we'll do in the break, which we will have shortly, is I will get you to select, and it's basically a first come, first select. So Mustafa beat everybody for lemons. The lemons are off the list. All right, guys, online, did you get that you have to select a crop? So in a minute, we're going to have a break, and I'm going to put up the, I'm going to share the assessment for you. I'll just minimize this and pull it up. And then you can have a look, and you can, both of you can start selecting what crop you might do your assessment on. All right?
Yep. And what's with the DIY kits?
Sorry, could you speak up?
What is with the DIY kit? So we can.
No. This is. I'll open it in a sec. Here we go. Oh, I have got more here.
Like I don't have my DIY.
You see this?
Yep.
This is what I'm going to put on Moodle for you today, which I've just handed out, and it's details about another assessment for this for this subject. You, as part of this assessment, the first task you have to do is select a theoretical crop, and there are a number of options on this list, and we can add others. The crop that no one can have is wheat. All right, it's deliberately not on the list. Okay, or it shouldn't be on the list. If it's on the list, take it off the list. Because I use wheat as an example to take you through what a crop management plan would look like from a detailed perspective. So I don't leave you out and dry. We don't have enough time in this course to cover every single crop, and it wouldn't be wise, because you know, to be able to grow every single crop in every single area of Australia under every single different parameter would take many lifetimes of farming to get that kind of knowledge. But what we do intend to do is give you the foundations so that you can go away and adapt your basic foundation knowledge to the specifics of wherever you may end up. So that's the objective here. Is that okay? All right, so I'm going to put this on hold for a second. How do I reduce this? And I'll just share that list with you, okay?
Nikki,
Right, I'm just going to pause the recording as well.